Welcome to our educational webinar titled Optimized Workflows for High-Efficiency Genome Editing in STEM and Primary Cell Types. My name is Alexander Schlacht, and I am a product manager at STEMCELL Technologies. I'll be your moderator for this webinar. If you'd like to ask a question during the presentation, please use the Q&A panel or chat box on the right side of your screen, and we will address those questions at the end of the presentation. It is now my pleasure to introduce you to our speaker today. Dr. Ashley Watson is a scientist at STEMCELL Technologies. Ashley received her PhD in biochemistry and developmental biology from the University of Western Ontario in 2015, where she focused on the regulation of chromatin structure neurodevelopment. She then completed her postdoctoral training at MIT, where she used CRISPR-Cas9 to generate novel 2- and 3-dimensional cell culture models of neurological disease from isogenic human-induced pluripotent stem cells. At STEMCELL, Ashley is currently working to optimize genome editing in diverse cell types and oversees the genome editing and molecular tools product lines. Let me now hand you over to Ashley to begin the presentation.
Okay, thank you, Alex, for that introduction. I am very excited to be here today. Thank you all for tuning in. I'm going to be telling you about some optimized workflows that we've developed here at STEMCELL Technologies to obtain high-efficiency genome editing in STEM and primary cell types. So I'm going to begin this presentation by giving a brief overview of CRISPR-Cas9 and its use as a genome editing tool. And then I'm going to go into how it's being applied in more complex culture systems such as STEM and primary cell types, and some of the challenges that are associated with applying this tool in these complex culture systems. And then I'm going to go into some specific examples related to editing in human primary immune cells and human pluripotent stem cells, and how STEMCELL Technologies is approaching these challenges in applying genome editing in these cell types. So genome editing as a field has actually been around for quite some time, and only recently has it been really propelled into mainstream use, and this is particularly due to the CRISPR-Cas9 system and its increased feasibility over more traditional genome editing methods. So instead of relying on protein DNA recognition like zinc finger nucleases and talons, targeting of the Cas effector nuclease is based on Watson-Crick base pairing between a guide RNA and the target DNA. So the simplicity of guide RNA design for new targets, the need for only a single constant protein, so you don't need to engineer proteins, which is very costly and labor intensive, as well as the modest cost and complexity of the CRISPR-Cas9 platform has really made targeted genome modifications readily accessible to researchers around the world. One thing I want to highlight is that regardless of the technique, all genome editing relies on DNA damage. And so most of the time, this type of DNA damage is a double-stranded DNA break that's induced by the nonspecific nuclease, such as Cas9. And then from there, endogenous double-stranded break repair mechanisms within the cell will take over. And it's really the outcome of these repair events that dictates genome editing outcomes. So the mechanism of how CRISPR-Cas9 works is as follows. As I mentioned, there's the Cas effector protein, in this case is Cas9, shown here in this bean shape, which pairs with its targeting molecule, the guide RNA, shown in blue. So the guide RNA is made up of two parts, the CRISPR RNA or CRRNA and the tracer RNA. The tracer RNA is complementary to the 3' end of the CRISPR RNA, and once annealed, these two components form a binding scaffold for interaction with the Cas9 protein. Probably the most important part of the guide RNA is the 5' end of the CRISPR RNA, which is usually 17 to 24 nucleotides and most often 20 nucleotides complementary to the target region of the genome. Another important part of this system is the PAM site or protospacer adjacent motif. And for Cas9, this is NGG, where N is any nucleotide. And it must be immediately downstream of the targeting sequence. So once Cas9 pairs with its guide RNA, it'll scan the genome for these PAM sites, and when it identifies a PAM site with a complementary upstream region that's similar to the CRISPR RNA sequence, It will unwind the DNA and interact with it, and Cas9 will generate a double-stranded DNA break, 3 to 4 nucleotides or base pairs upstream of the PAM site. And so the exact place that Cas9 cuts is very well defined. Once Cas9 generates this double-stranded break, as I mentioned, endogenous DNA repair pathways will take over. So there's 2 main double-stranded break repair pathways in mammalian cells: non-homologous end joining and homology-directed repair. Non-homologous end joining, or NHEJ, is the predominant pathway in most cells, and it acts to directly ligate broken DNA ends back together. So typically this is not very error-prone and most of the time results in repair with high fidelity. However, in the context of CRISPR-Cas9, high-fidelity repair will result in wild-type sequence, and then the guide RNA and Cas9 can bind and continue cutting until imprecise processing disrupts the CRISPR RNA targeting sequence or the PAM site. And so this imprecise processing of the broken DNA ends can result in insertions or deletions or indels. And this is very useful in the context of genome editing because it can enable functional gene knockouts if these indels generate a frameshift mutation in a gene. Alternatively, you can also use this method to delete regulatory elements and study enhancer function or promoter function. The alternative double-strand break repair pathway is known as homology-directed repair. And this occurs actually at quite a low frequency in the majority of mammalian cells. And the reason for this is that it's typically active in late S or G2 phase after DNA replication. And so in the context of normal biology, after the DNA has replicated and there is a sister chromatid that's homologous to a site that might have a double-stranded break, then that sister chromatid can be used as a template for repair. In the context of genome editing, the user can incorporate an exogenous donor DNA that can act as a template for precise knock-in of small point mutations or large gene insertions. So this template DNA will have homology arms spanning the target site, as well as incorporation of whatever kind of change you want to make in the genome. And so this really powerful pathway enables precise rewriting of the genome and allows for disease modeling. So you can correct or introduce patient-specific mutations using this method. Alternatively, it's also useful for repairing genetic disorders. So if we know a specific mutation that is causative for a disease, potentially this method can be used to repair that monogenic disease. So traditionally, or I guess in the early days of CRISPR-Cas9 as a genome editing tool, this tool was applied in immortalized cell culture systems. And so these kinds of cell culture systems are, have acquired a non-physiological ability to proliferate indefinitely. And this is because they're typically isolated from tumors, or they can be immortalized with viruses, and this allows them to propagate indefinitely in vitro. So they're very useful because they're very easy to maintain, they're easy to propagate, and they're very easy to manipulate as well. However, there's also some drawbacks with these immortalized cell lines, including that they do not very closely mimic in vivo conditions, and they can harbor several chromosomal aberrations, and this is because they're typically cancer cell lines. So some examples include HeLa cells, A549, and HEC293. So alternatives to these cell culture systems that better resemble the natural state of an organism include primary cells, which can be freshly isolated cells from tissues of the host species, or induced pluripotent or embryonic stem cells, or human pluripotent stem cells, as we like to refer to them at STEMCELL. So primary cells, as I mentioned, are isolated from individual donors. They very closely mimic in vivo conditions because they're taken directly from a patient and put into, or directly from a donor and put into culture. And they have very minimal chromosomal aberrations because of this. Some examples include T cells, hematopoietic stem and progenitor cells, and also tissue biopsies. So you can take, for instance, intestinal biopsies and culture them in 2 or 3 dimensions from human tissue. Some drawbacks of primary cells is that they can be very complex and difficult to culture. As well, they exhibit very limited cell division in vitro, and this can be a significant challenge, especially when you're trying to edit these types of cells. When primary cells aren't available, so in the case of if you want to model a cardiac disease or a neurological disease, researchers have turned to human pluripotent stem cells. So these types of cells are a very powerful tool. They can be isolated from individual donors, either skin cells, blood cells, etc., and then reprogrammed into stem cells. And because they are now pluripotent stem cells, they exhibit indefinite cell division in vitro. They also, since they come from human donors, they closely mimic in vivo conditions. And they can exhibit most of the time very few chromosomal aberrations. However, some chromosomal aberrations can occur with long-term passage, and I'm going to touch upon this point in subsequent slides. So some examples of IPS lines and ES lines, there's been thousands that have been generated from a variety of different donors, from both healthy individuals as well as individuals suffering from disease. And importantly, these cell types can be differentiated into really almost any cell type in both 2- or 3-dimensional cultures. And there's constantly more and more protocols and kits out there to differentiate pluripotent stem cells into different cell types. One drawback of pluripotent stem cells is that they can, again, like primary cells, be complex and difficult to culture. But together, primary cells and human pluripotent stem cells represent really exciting tools, especially when you apply CRISPR-Cas9 genome editing tools to these culture systems. As I mentioned, despite the fact that they can be very exciting and powerful tools for disease modeling and for studying basic biology, there's still some significant challenges when editing stem and primary cell types. The primary challenge that's common between both stem and primary cell types is that they are inherently more challenging to culture than immortalized cell lines, and they are very challenging to manipulate as well. These cell types require a specialized medium for their growth, and so PSCs in particular typically require daily feeds, and this can be a significant pain point when working with this type of cells. They're almost like newborn babies, and they need really, really attentive care in order to maintain them in culture. Primary cells, in contrast, are very sensitive to changes in culture conditions, and again, they only undergo a finite number of divisions. And this is because they are directly isolated from a donor, and so you need to really carefully consider the culture conditions in order to fully support the growth and expansion of these cells. Secondly, there are challenges in delivering and expressing the CRISPR components in both stem and primary cell types, but particularly in primary immune cells, they are immune cells. They have an innate mechanism to resist foreign genetic material, and so you have to choose the format of how you're going to express the CRISPR-Cas9 system in these cells very carefully to minimize cytotoxicity. Both of these types of cell culture systems exhibit low clonogenicity and single-cell survival. This is particular for pluripotent stem cells. And so derivation of clonal cell populations can be quite challenging in these different cell types. And finally, pretty much all mammalian cells are quite sensitive to DNA double-stranded breaks. And as I mentioned, all genome editing relies on DNA damage. And so the DNA damage response can hinder editing efficiency. And so throughout the next several slides, I'm going to go into all of these challenges and how we're trying to address them here at Stem Cell Technologies. So first, I'm going to discuss a little bit further the format and of expressing CRISPR-Cas9 components in these cell types, in these cell systems. So this is a schematic just showing the different expression and delivery methods that currently exist. So this is an image of a cell. This is the cell membrane. Oops, sorry. The nuclear membrane here. And so you can see the three main systems are DNA by expression plasmids, mRNA, or ribonucleoprotein complexes. And so if you want to express CRISPR-Cas9 on an expression plasmid, you can encode the Cas9 gene with the guide RNA and deliver that plasmid into the cell where it can be transcribed and translated into Cas9 protein and guide RNA that will form the ribonucleoprotein complex. and then enter the nucleus to undergo editing. Alternatively, you can directly inject Cas9 mRNA with guide RNA into the cell, which will then be translated into the complex for genome editing. And finally, the method that's become very popular for more complex cell types like stem and primary cells is ribonucleoprotein, wherein you can take purified Cas9 protein, pre-complex it with synthetic modified guide RNAs, and then you can directly deliver the ribonucleoprotein complex into cells where it can immediately act and perform genome editing. And so this method has become very popular, first because it overcomes any cytotoxicity associated with delivering foreign nucleic acids into the cell. And there's a few additional features of the RNP method that make it beneficial over some other systems. So here's some data showing enhanced performance of a protein-based CRISPR-Cas9 system compared to DNA and mRNA, wherein across electroporation conditions, you can see that the indel production or editing efficiency is higher for protein than the other systems. And one of the most key pieces of data related to this is that the RNP system exhibits very transient expression. So you can see that after delivery of the RNP, that the Cas9 expression, this is a Cas9 Western blot, peaks at around 4 to 8 hours and is almost gone and undetectable by 72 hours in the cells. However, this is in stark contrast to when you deliver CRISPR-Cas9 on an expression plasmid, where you can see that Cas9 expression peaks at around 24 hours and is still very high even after 72 hours of delivery. And so this indicates that Cas9 is still present in the cell and can result in higher off-target effects because it's kind of lingering in the cell. And this is very nicely reflected in this data here, measuring off to on-target ratio at 2 off-target sites for a given guide RNA. And you can see that with the plasmid or DNA-based system, the off to on-target ratio is highest, especially compared to RNP or protein-based systems. And so here at STEMCELL, we offer the Architect CRISPR-Cas9 system, which is an RNP-based system. And this is what we feel is the best system for editing stem and primary cell types because of the data I just showed you. So this is a general workflow that we've developed at STEMCELL for editing really any type of cell type. So first you need to design your experiment, design the CRISPR RNA and guide RNA. And then following that, you need to optimize your culture conditions. And we feel that pre and post-editing culture optimization is a very key aspect to focus on when editing these complex cell types. Next, you form and deliver the RNP complex. You allow editing to occur afterwards, and then you can analyze and characterize your cells. And there's several different methods out there. I'm going to touch upon some of them, including sequencing, T7 endonuclease, or flow cytometry, and it really depends on your experimental design as to which is the best characterization method for you. So now I'm going to go into a specific example of how we optimized a workflow for editing human primary immune cells. So as I mentioned, when editing human primary immune cells or human primary T cells, there's some critical considerations. So the first consideration is the delivery and expression format of the CRISPR-Cas9 system. And so in these cells, the most optimal method is to couple purified Cas9 protein with chemically modified guide RNAs of high purity. And our Architect CRISPR-Cas9 system is this type of system. So beyond CRISPR-Cas9 expression methods, the systems for isolation, expansion, and activation of primary human T cells also represent critical elements for successful genome editing. And so while T cells can be isolated from a number of sources using a variety of isolation techniques, most methods to date involving T cells have used primary human T cells that are isolated from peripheral blood mononuclear cells by immunomagnetic selection, and that's what's shown here. So you can use our EasyStep immunomagnetic selection kits to isolate T cells of very high purity. You can then culture those cells activate them, and then deliver the RNP. And then we have optimized protocols to expand those T cells, followed by analysis of editing efficiency. So a very exciting area of application for CRISPR-Cas9 in the immune cell space is to engineer chimeric antigen receptor T cells for cancer immunotherapy. And so traditionally, how CAR T cell engineering has worked is that you harvest T cells from a patient. You then transduce those cells with a virus that harbors the CAR construct, which will then be randomly integrated into the genome, and then you'll have T cells that express that CAR. However, because it's randomly integrated, it could integrate into a gene and disrupt that gene's function. And alternatively, over time, sometimes those randomly integrated CARs can be silenced. Nonetheless, this is becoming a really promising area for cancer immunotherapy. You can take those T cells, you can expand them, and then administer them back into the patient. However, now CRISPR-Cas9 is being rapidly applied to this kind of technology because it enables off-the-shelf, universal, allogeneic immune effector cells. And so now you can harvest T cells from either donors that are healthy or the patient, and then you can engineer those T cells to escape immune rejection by deleting the T cell receptors and HLA receptors, as well as deleting inhibitory receptors like PD-1. At the same time, these two strategies can be coupled with precise knock-in of the CAR, and often the CARs are targeted to the T cell receptor alpha locus, and this enables the CAR to utilize the endogenous promoter for TCR alpha for expression. So because of this, in order to develop our optimized methods, we chose to target the T cell receptor alpha locus. And so this is what the TCR alpha locus looks like, and we targeted exon 1, shown here, and then we designed 4 different CRISPR RNA or targeting portions of the guide RNA to test to see which one performed best at this locus. So in order to test this, we either mock electroporated the T cells, which express high levels of CD3 and TCR alpha beta, which is to be expected, or we electroporated the T cells with RNP complexes comprising CRISPR RNAs 1 through 4. And what we found was that while all of the RNP complexes with CRISPR RNA 1 through 4 all resulted in knockout of TCR alpha beta and CD3, which are co-expressed, and that's shown here, they actually knocked out TCR alpha beta to varying degrees. So although all of these CRISPR RNAs lie within a very similar region of the genome, there was varying efficacy, and we found that CRISPR RNA2 actually performed the best out of all the CRISPR RNAs. And so this just highlights that when applicable, testing multiple CRISPR RNAs is a really useful strategy to identify the CRISPR RNA that works the best, because even CRISPR RNA2 and CRISPR RNA1 are nearly identical except for a couple base pairs. And you can see that the efficacy of these 2 different CRISPR RNAs is significantly different. So we move forward using to optimize our workflow using CRISPR RNA2. So our general workflow is as follows. We isolated T cells using our EasySep negative selection kit, and then we activated those T cells with our immunocult T cell activators in culture for several days or a few days prior to delivering the RNP composing of CRISPR RNA2. We then cultured the cells for several days after, and then monitored knockout status by TCR alpha beta and CD3 flow cytometry. So initially, we cultured the cells with and without the presence of our immunocult CD328 activator. And you can see that in the presence of this activator, the T cells aggregate and exhibit an activated phenotype to be expected. We also monitored activation status by flow cytometry with the CD25 T cell activator marker. And you can see that in the presence of activator, almost 90% of the cells are CD25 positive, whereas in the absence of activator, only very few cells express CD25. We then delivered the RNP complex into these cells, and so in the mock electroporated cells, in both without and with the presence of activator, you can see that the level of TCR alpha beta and CD3 knockout is quite low. And then in the presence of activator, we saw a high degree of TCR alpha beta CD3 knockout, which was exciting. However, in the absence of activator, we saw very low levels of knockout, indicating that at least under these conditions, our immunocalled activator is required to obtain high efficiency editing. So moving on from there, we wanted to test activation dynamics, and we also sell a couple different immunocalled activators, so we wanted to test which one would be preferred in this kind of workflow. And so this data is shown here. So in the mock electroporated cells cultured either with the CD328 immunocalled activator or the CD328CD2 immunocalled activator, for 2 or 3 days in culture, we saw very low levels of knockout. So these cells still expressed very high levels of TCR alpha beta and CD3. And then in the cells that were electroporated with an RNP that had the CRISPR-RNA2 targeting sequence, cultured for either 2 or 3 days with either activator, we saw robust knockout of TCR alpha beta and CD3. The condition that we found, that was the best condition was the 3-day pre-culture activation with CD328 prior to delivery of the RNP, which yielded almost 90% knockout efficiency of TCR alpha beta and CD3 compared to the mock electroporated cells. And importantly, these cells, despite loss of TCR alpha beta, still express T cell markers such as CD4 and CD8. So now downstream of editing, while it's all well and good that we get very high editing efficiency, we also want to make sure that you can robustly expand these cells so you can get enough cells to perform downstream experiments. And so at STEMCELL, we've also optimized culture conditions post-editing. And we found that there is a strong effect of early cell dilution on the fold expansion of your T cells. So in these experiments, T cell culture volume was increased 2, 4, 8, 16, or 32-fold on day 3 of culture. As you can see, the 4- and 8-fold culture volume increases resulted in the highest total fold expansion, as well as the highest cell viability, without any significant effects on the activation phenotype of those expanded cells. So together, these data highlight the importance of cell density on T cell expansion, and our findings suggest that maintaining T cells at lower cell densities significantly improves T cell growth and viability, especially at early stages of cell expansion. So just a brief summary of genome editing in human primary immune cells. We know and we appreciate that primary immune cells can be very challenging to manipulate. And this is due to the fact that delivery of exogenous nucleic acids can result in very high cytotoxicity, especially in these types of cells. And so our Architect CRISPR-Cas9 RNP-based system really supports high-efficiency genome editing in this type of cell. And we've also identified some other key areas for optimization in the cell type, including pre-editing immune cell activation that might be necessary for efficient editing, as well as the fact that cell density is a key factor for robust expansion of human primary T cells. Now, as I mentioned previously, in primary cells, they have very low clonogenicity. And so depending on what kind of editing outcome you want, this high editing efficiency that we were able to establish might be able to overcome these limitations. So if you're looking to knock out a certain gene, because we can see efficiencies of around 90%, this may be able to overcome these limitations. However, the generation of clones with a specific genetic change or with a knock-in still remains a challenge. And there's groups that are actively working to try and address this challenge, including identification of alternative templates for non-viral knock-in. So to knock in the CAR, especially if you want to take these cells downstream into the clinic, you most likely want a non-viral strategy. And very recently in Nature, Alex Marson's group published, Roth et al., this strategy of using long single-stranded DNA that incorporates the CAR into T cells. And they see relatively high on targeting efficiency. However, as you can tell, it's still around 10%. And so there's still a significant area of improvement here. Alternatively, if you want to obtain a clonal population of cells, a strategy that you might be able to employ is that you can edit human pluripotent stem cells. And because these cells, although still challenging to clone, it is possible to clone them, you can generate clones with a specific genetic change and then differentiate those clonal cells into T cells. And we're actively working on a kit in order to accomplish this. But that brings me to the second part of my talk, which is focusing on optimized workflows for genome editing human pluripotent stem cells. So I'm going to go back to this challenges slide and just highlight some of the stem cell specific challenges that are present here. So as I mentioned, stem cells are very challenging to culture and maintain, and one of the main reasons for this is because they typically require daily feeds. However, very recently, we just launched a new media for pluripotent stem cell maintenance known as mTeaser Plus. So this is based on the mTeaser formulation. We've improved it in two different aspects. So one of the reasons why you need to feed pluripotent stem cells daily is because some of the key pluripotency factors are not very stable at 37 degrees, including FGF2. And so over time, at around 24 hours actually, there's usually very negligible levels of FGF2 left in the media. And so this is one of the reasons why you need to continually replenish the media. Another reason is that because pluripotent stem cells have a very high metabolism, this can result in medium acidification. And so the two features that we've improved in mTeaser Plus is that we've stabilized FGF and we've also enhanced the buffering capacity of this media to try and prevent or counteract this medium acidification. And so the data showing this is here, where with your traditional mTeaser 1, the level of FGF2 that remains after about 24 hours is almost negligible, whereas with mTeaser Plus, the level of FGF2 is pretty constant up to 72 hours in culture. Importantly, because of our enhanced buffering, compared to an alternate media and mTeaser 1, which show reduced pH or acidification of the medium, especially with increasing cell numbers after 3 days, we see enhanced buffering in mTeaser Plus that's shown in this orange line. So that's addressing that one pain point, but there's a few other pain points related to the PSC genome editing workflow. And so this is the workflow we've developed where, again, you design your CRISPR RNA, you form and deliver the RNP complex, and we've worked out the exact amounts of Cas9 and guide RNA, as well as donor DNA template in the form of single-stranded oligodeoxynucleotides to deliver into the cells. And then we've optimized post-editing culture conditions so that you can derive clonal cell lines and then establish those lines. And so I'm going to go through each one of these stages step by step in the subsequent slides. So in order to develop this optimized workflow, we utilize the GFP to BFP conversion assay, which enables quantitative assessment of editing efficiency. And so this is because the GFP gene differs from BFP by 2 different nucleotides. And so you are able to target this region of the genome using a CRISPR RNA sequence to GFP. And then you can deliver a single-stranded donor DNA template that's essentially identical to the GFP gene, except contains the nucleotides that convert it to BFP. And we also incorporated some synonymous mutations shown here in red to prevent guide RNA reannealing and some break cycles. And so with this strategy, we utilized some in-house induced pluripotent stem cell and embryonic stem cell lines that have a GFP cassette. And so we took those GFP expressing lines and we electroporated them with RNP that had the CRISPR RNA targeting GFP and then co-delivered with the donor DNA template shown here. We then cultured those cells for 72 hours and then assessed editing efficiency by flow cytometry. And so here you can detect wild type or GFP positive cells. You can also detect editing events in the terms of knockout or GFP negative cells and knock-in or BFP positive cells. So this is an example of what those FACS plots look like, wherein mock cells express high levels of GFP. If you only deliver the RNP, you can see a pretty efficient knockout at around 50 to 55%. And then when you incorporate co-delivery of the single-stranded DNA template, you can see knock-in of BFP. And so we wanted to test whether, we wanted to test editing in these pluripotent stem cell lines using either pre-culture with mTESR plus, mTESR 1, or an alternate media that claims to be specifically designed to support genome editing in pluripotent stem cells. So as you can see, across the different media culture systems, in terms of knockout and knock-in efficiency in two different pluripotent stem cell lines, there's really no difference in editing efficiency. And so all of these media systems support high-efficiency genome editing in these two different cell types. One thing we noticed is that there is a clear relationship between growth rates and overall editing efficiency. And so we recommend that you collect the cells during the exponential growth phase in order to perform editing. And this is typically when cells are most amenable to uptake of foreign cargo, such as the RNP. So next, after you edit the cells, what you have is a heterogeneous population of cells or a genome edited cell pool. And so these cells, the cell pool can be really useful if you want to study gene function. Again, it can be similar to an RNAi knockdown experiment to study the function of a gene by knocking it out. But alternatively, this genome edited cell pool is actually really useful to assess editing efficiency, and it can be very informative in clonal derivation. And so I'm just going to describe how you might go about doing this. So you can take the genomic DNA from the parental line as well as your genome edited cell pool. You can PCR amplify the target region and then send it for Sanger sequencing. And what you'll get back is something that looks like this. So for the wild type or parental line, you should get a clean trace. Whereas for the mixed genome edited pool, you'll at the cut site, you'll start to see all of these different traces representative of all of the different editing events that occurred in that mixed population. So a few years ago, this group published the TIDE program, which actually will take these mixed traces and deconvolve them. And it gives you a relative proportion of the different alleles that are present within your population. And so this can be very informative of how many clones you might need to select for further characterization. So for instance, if you wanted a clonal population that contained the +1 edit, then you would know that about 1 in every 10 clones should theoretically have that edit. And there's been subsequent studies that show that this method is actually really strongly correlated with next generation sequencing or targeted next generation sequencing. And so it can be pretty useful in identifying how many clones to select. So then you can, once you know that, let's say you want to select 20 clones, you can single cell sort those clones into a 96-well plate, or you can plate them at very low density in order to derive clonal pluripotent stem cell lines. But again, this part is a significant pain point because pluripotent stem cells are quite challenging to clone, and they exhibit very low single cell cloning efficiencies. So we know that genome editing is a stressful process. It involves electroporation typically and delivery of the RNP complex. It then involves a double-stranded break that the cell needs to overcome and repair, and hopefully repair in the way that you are intending. And so it places a high degree of selective pressure on pluripotent stem cells. So there's a number of bottlenecks that restrict single-cell survival of pluripotent stem cells, especially in the genome editing workflow. So after you take single cells and you plate them, they need to survive that plating. Then they need to re-enter into the cell cycle and survive that division, and typically this results in cloning efficiencies of less than 1%. However, a few years ago, we developed this product called Cloner, which is a supplement for mTESR1 and mTESR Plus that enables robust generation of clonal pluripotent stem cell lines. So in contrast to Rocky, which is a traditional small molecule that helps support survival of pluripotent stem cells, Cloner robustly supports survival of these single cells, and that's shown in this slide here. So yeah, compared to Rocky across different stem cell lines, you can see that there is much more robust cloning efficiency with Cloner supplementing mTESR1 or mTESR Plus. And one thing to note is that with mTESR Plus, if you supplement it with Cloner, that the colonies that you start to see appearing actually are much larger at the same day as with mTESR1. And so in a genome editing workflow, this can be really useful because you might be able to pick your single cell clones a little bit earlier. So then once you've derived your clonal pluripotent stem cell lines, you then want to establish and characterize those lines. So again, I just want to reiterate the fact that editing is quite a stressful process, and it places a high degree of selective pressure on pluripotent stem cells. And so because of this, it's very important to ensure the quality of your cells is maintained after editing and during long-term maintenance. So there's some areas of cell quality that we have identified, including morphology of the pluripotent stem cells. You also want to make sure that your cells are still pluripotent in that they can differentiate into all three germ layers and that they express markers of the undifferentiated state. Finally, you want to ensure that your pluripotent stem cells maintain genetic stability. And we think this is highlighted here because we think that this is a very important consideration for pluripotent stem cell quality during maintenance culture and especially after editing, because as I mentioned, all genome editing relies on DNA damage, which can result in genetic instability. And so shown here is a representative karyogram of one of the most common chromosomal aberrations that are observed in pluripotent stem cells during long-term culture, which is triplication of chromosome 12. And so this can result in selective advantage for the cells, and those cells can overtake the wild type cells. And then why this is important and why we think you should really be considering this is because if you, at the end of your project, get all this really interesting data comparing a control line to your edited or mutant or patient line, and then you go to karyotype the cells when you're trying to publish and you find that your mutant line actually harbors a chromosomal aberration, well, then all of your data is called into question because you're not sure really if the phenotype that you're observing is due to this change that occurred during maintenance culture, or if it's due to the edit that you made or the actual genetic change that you're trying to study. Furthermore, in the context of genome editing, genetic stability is really critical because in 2018, 2 groups reported on the phenomena that the p53 pathway is activated by CRISPR-Cas9 induced double-stranded breaks, and that this response reduces the efficacy of genome editing. Importantly, this indicates that p53 status should be carefully monitored before and especially after you edit your cell lines. And this resulted in even more popular scientific reports linking potentially CRISPR-edited lines to cancer risk, which took it a little far, but it kind of makes a bit of sense because if you think about it, when cells have a functional copy of p53 and then you deliver, for instance, an RNP complex and Cas9 creates a double-stranded break at your target site, p53 becomes activated. It then activates its downstream effectors such as p21. And the possible outcomes of this are apoptosis or cell cycle arrest. And often these are the 2 outcomes, especially in very sensitive cell lines like pluripotent stem cells. Alternatively, the other outcomes are that there's no break that's formed, or that repair is, there's repair with high fidelity, or you can get efficient genome editing. However, if you have cells that don't have functional p53, then p53 does not become activated. There's no apoptosis or cell cycle arrest, and so there's a higher proportion of cells that undergo efficient genome editing. However, you don't want to be studying cells that have a non-functional p53. And if you ever want to take those edited cells into the clinic, you definitely don't want to be transplanting cells that have non-functional p53, as these cells are often tumorigenic and can cause cancer in the donors. And so this is something really critical to monitor before and even after editing. Furthermore, there are several recurrent genetic abnormalities that can occur during long-term culture. Chromosome 12, whole chromosome gain of chromosome 12 is one of them that I had touched upon earlier. But this is an ideogram showing all of the different chromosomes in the human genome. And this study looked at long-term pluripotent stem cell cultures and the types of chromosomal aberrations that they acquire. So you can see that there, like chromosome 12, there's other chromosomes that have recurrent gains or recurrent losses or rearrangements. And I want to highlight one of these because it's particularly relevant to genome editing, and that's chromosome 20. So there's a region on chromosome 20, it's chromosome 20q 11.21, that actually shows recurrent gains in human pluripotent stem cells, especially when they're cultured long-term as single cells. So this 20q amplification is associated with enhanced pluripotent stem cell survival. And this study that came out a few years ago showed actually that within this region of 20q, the BCL-XL gene is actually the gene that is responsible for this enhanced survival. And so interestingly, BCL-XL actually lies within the p53 pathway. It's an anti-apoptotic protein that's actually repressed by p53, and it itself represses BAX and BAC, which are pro-apoptotic factors. And so when you overexpress BCL-XL, either because you have 20q amplification or by other means, then this would result in reduced apoptosis. And so importantly, in the context of genome editing, this study came out last year showing that overexpression of BCL-XL does improve cell survival in pluripotent stem cells, and it also significantly improves knock-in efficiency at 3 different loci in these pluripotent stem cells. And so while potentially transient overexpression of this anti-apoptotic factor can be beneficial for genome editing, if you have cell lines that exhibit 20q amplification, you might be seeing really high editing efficiency, even though you also have these additional genetic aberrations. And so last year, we launched a qPCR-based kit known as the HPSC Genetic Analysis Kit, which contains primer probe sets that cover 75% of the most recurrent culture-dependent abnormalities in human pluripotent stem cells, including primer probes to detect 20q amplification. And so interestingly, as you can see here in this karyogram, even though this cell type, as we detected using the qPCR-based kit, has 20q amplification, by traditional karyotyping methods, you can't detect it because that 20q region is too small. But this was validated using FISH. And so this kit, we feel, is a really useful kit to especially pre-screen edited lines for any abnormalities prior to sending them for karyotyping or performing any downstream analysis on these cells. So I'm just going to finish off by highlighting an exciting application for use of edited human pluripotent stem cells, and that's in disease modeling. So you can take induced pluripotent stem cells from healthy patients, and you can edit those cells to harbor disease-associated changes. And then you can differentiate those cells into disease-relevant cell types and compare the phenotype between these isogenic lines. Alternatively, or in parallel, you can take patient-specific iPS cells, edit them to correct the mutation, and then again compare the phenotype. And so this overcomes a lot of the variability that's associated with pluripotent stem cells from different donors. And this allows you really strong models of disease, especially when you can differentiate these cells into cell types that are not readily accessible, like neurons or cardiomyocytes. And so we performed a project at STEMCELL to model microcephaly using human pluripotent stem cells that were edited to harbor a associated mutation, or a mutation that's associated with microcephaly, and then differentiated those pluripotent stem cells into cerebral organoids with our STEMDiff kit. So in orange is the CRISPR RNA targeting sequence in the CDK5-RAP2 gene that we targeted, and we obtained isogenic pairs of both control, heterozygote, and compound heterozygous lines using our architect system. We then monitored the quality of these cells by both morphology, markers of the undifferentiated state, as well as pluripotency using our trilineage differentiation kit. So this kit is a 2D monolayer based directed differentiation kit that allows you to assess whether your cells can differentiate into ectoderm, endoderm, or mesoderm. And so using these quality control measurements, we found that the cells looked very good between the different edited lines. We also found that by karyotype and genetic analysis kit methods that those cells were also genetically stable. So then we assessed the level of CDK5-RAP2 in these different lines, and we found that heterozygotes and controls still expressed CDK5-RAP2, whereas the compound heterozygote did not express CDK5-RAP2, and it did not have its traditional localization at the centromere. We observed similar growth curves between these cells when they're at the pluripotent stem cell stage. And then when we differentiated those cells into cerebral organoids, we noticed a significant phenotype in the compound heterozygote where they did not grow as large over time as their control or heterozygote counterparts. And then we found that this growth difference was associated with altered differentiation of those neural progenitor cells into neurons, and that the compound heterozygote had increased neuronal differentiation at the expense of progenitor proliferation. And so this short study just highlights the utility of applying genome editing tools like our architect system to pluripotent stem cells, and then using our really well-established kits to differentiate those cells into a specific disease model, and then you can study a disease phenotype. Okay, just to summarize everything I've said today, we feel that there's some key considerations to obtain high-efficiency genome editing in stem and primary cell types. The RNP system is currently the best available method to support genome editing of these cell types. It's the most efficient, it allows direct delivery of the RNP complex, and it has the lowest chance of off-target effects. And we also feel that there's careful consideration of pre and post-editing culture conditions is required in order to obtain high-efficiency editing. And so in primary immune cells, it appears that activation of those cells does promote efficient editing, and that low-density cultures promotes post-editing cell expansion. In pluripotent stem cells, it's critical to collect and edit cells during their exponential growth phase, that robust media systems like mTeaser, mTeaser Plus supplemented with cloner is critical to support post-editing survival, and that it's really important to monitor genetic stability during long-term culture and after editing, and that can really help to support your editing experiments. So with that, I'd just like to thank you all for listening, and I'll take any questions. Oh, and I'd just like to acknowledge, all of the people that helped me with this work, including the Genome Editing and Molecular Tool group here at STEMCELL, the Immunology group, the PSC group, and the Neurobiology group. And this is an image of where we're located, which is in Vancouver, Canada. And we're always looking for really qualified individuals to come work for us. So this is just a quick plug showing how beautiful Vancouver is. Thank you.
Thank you very much, Ashley. That concludes the presentation segment of the webinar. We'll now begin a Q&A session by addressing the written questions that were submitted throughout the talk. As a reminder, if you do have any questions, please submit them using the chat box on the right, and we'll pass those to Ashley to be answered. So for our first question is, where exactly does the guide RNA cut?
So the question was, where exactly does the guide RNA cut? The guide RNA actually doesn't do any of the cutting. It's just targeting Cas9 to the genome. But Cas9 will generate a break between 3 and 4 base pairs upstream of the protospace or adjacent motif. So it's the 3 to 4 base pairs at the end part of the targeting or CRISPR RNA sequence of the RNA.
Our next question is is the RNP preparation step easy or difficult? What does that look like?
Yeah, so the RNP preparation step is actually quite straightforward. So in our system, we have a 2-part CRISPR RNA and tracer RNA system, which involves a pre-annealing step that can be performed in a PCR machine, and it takes about 10 minutes, or I think about 6 minutes actually. And then to form the RNP, you take the annealed guide RNA and you mix it with the Cas9 protein, and that you let it incubate at room temperature for between 10 and 20 minutes. So it's pretty straightforward.
Our next question is, why do you use electroporation instead of other delivery methods like chemical transfection or cationic complexes?
Yep. So we have tested, at least in pluripotent stem cells, several chemical-based transfection methods, but by and large, we find that electroporation is the most efficient method to deliver the RNP complex, and so that's why we chose electroporation. And the same goes for primary immune cells and really any other cell type we've tested at STEMCELL here.
The next question is, in primary immune cells, why do you think the activation step is important for editing efficiency? Do you have any idea what might be going on mechanistically?
Yeah, so I don't know exactly, but I can speculate that the activation step promotes cell cycling. And it's well known that for cells to take up foreign cargo, that when they're actively cycling, they're just more readily or more amenable to take up that foreign cargo. So I think it has something to do with that.
And then on that vein, what about non-proliferating primary cells? Is activation sufficient, or is it really the proliferation of the cells that's needed?
Well, I think in the field, it's believed that proliferation of the cells can at least enhance delivery. However, there is some indication in the literature that activation is not always necessary, especially in primary T cells, in order to obtain high efficiency editing. And it might actually be that you need to optimize your delivery parameters, so the electroporation conditions, in unactivated versus activated cells. That might help.
Our next question is, iPSC generation sometimes uses p53 suppression. Is this being looked into with CRISPR editing?
Yeah, so actually some of the initial publications performing CRISPR-Cas9 editing in pluripotent stem cells did use p53 inhibitors. And it does improve editing because when you inhibit p53, like I showed in that p53 pathway diagram, it will suppress cell cycle arrest and apoptosis. However, usually p53 is inhibited using small molecules, and we don't really know the consequence of global p53 inhibition, especially even if it's just for a short period of time. And so people do use it as a method to enhance genome editing, but we're not really sure of the consequences of that on global genomic stability in those cells cultured for the long term.
Do you have any recommendations for editing and culturing primary cell lines like fibroblasts in clonal populations, as many of them have low survivability of single cells?
Yeah, I haven't done those experiments myself, and yeah, I know it is quite an issue, especially with primary cell types like fibroblasts that cannot proliferate indefinitely to get clonal populations. And so because this is a significant pain point, hopefully in the future we can try and address that.
Most of your experiments showed editing in human T cells. Does CRISPR editing perform similarly well in murine cells?
I would assume that it seems as though, looking at the literature and going to different meetings, it seems like CRISPR works quite well across all cell types. The challenge is really in delivering and expressing the CRISPR components in a really effective way. And so in primary urine epithelial cells, I think it's just a matter of trying to optimize the methods of culturing those cells, delivering the CRISPR components. So theoretically, it should work. We don't have a protocol for that, but if there is significant need, we'll try and develop one.
So our next question, when looking for off-target editing, do you recommend focusing on the areas of the genome that are predicted to be off-target sites, or is there a need to worry about other sites that are not predicted to be off-target?
Yeah, so I think there still is no consensus in the field on what is absolutely necessary, and I think it really depends on what you want to do with the cells downstream. If you want to go, I guess the easiest method to test for off-target effects is to look by Sanger or targeted next-generation sequencing at those top predicted off-target sites, and that's really the best we can do as a field right now in predicting off-target sites. If you want to take those edited cells into the clinic, it will be necessary to perform a more global, unbiased analysis. So often you can do this using targeted next-generation sequencing methods like GuideSeq or BLESS, and these are methods to really detect where a double-stranded break has occurred in the genome, and so where Cas9 has generated an off-target cut. But if you're looking to use these cells for research use only purposes in modeling a disease, I think what is now the consensus is to generate multiple independent clones of your edit of interest, or alternatively, if you have a system where you can obtain both patient disease-specific lines and control lines, and you can both introduce and correct the mutations into independent lines and show a similar phenotype, then that should be sufficient in order to mitigate the effect of any off-targets. Also, you can use multiple guide RNAs in individual clones to show a similar effect using different guide RNAs, which also reduces the likelihood that your phenotype of interest is coming from an off-target effect.
So our next question, in plasmid-based systems, is there any data or evidence to suggest that Cas9 on its own might have any long-term effects on stem cell populations, renewal, differentiation, or just overall stemness?
Right. I don't believe there's any data specifically on long-term expression of Cas9 in pluripotent stem cells and its effect on downstream differentiation. But the longer that Cas9 is around in the cell, it could possibly interact with the genome and generate a break. It is a nuclease, so it can cut DNA. And so long-term expression of Cas9 is likely going to impact the cell in some way or another, but I don't think there's any specific data showing that phenotype.
Our next one is, do you have any other advice for single-cell clonal expansion of human embryonic stem cells?
I guess, so when you try to generate a single cell suspension of pluripotent stem cells and plate it at very low density, even without genetic manipulation, so without delivering an RNP and performing genome editing, the survival of those cells without the use of cloner or Rocky is very low, almost negligible. So I don't think that there's any way without using some sort of supplement to support survival of those single cells to perform single cell cloning in PSCs.
Another question related to electroporation, which system do you use?
So for our studies, we primarily use the Neon electroporation system. It's really useful because you can go down to really low volumes and it's scalable. We also use the Lonza nuclear effector system, which is also a really efficient system for delivering the Cas9 RNP. And on our website, we have, in our technical bulletins, we've outlined instructions for how to deliver the RNP complex with the Neon system. And you can contact me or our product and scientific support team in order to get instructions on how to perform delivery with the Lonza nuclear effector.
Our next one is whether or not you have any experience targeting or editing intestinal organoids, and if so, what considerations might you suggest for targeting the stem cell component through multiple stem cell types?
Right. That is a very interesting question. This is something that we're actively investigating because it is a very useful system in order to edit adult stem cells, particularly adult intestinal stem cells, and generate edited organoids from those edited cells. It's an area of active investigation. If you wanted to get into contact with me, I can try and give you some initial suggestions for optimization. But yeah, this is an area we're actively working on. So hopefully look out for some more instruction in the future.
I think since we're running out of time, let's be our last question is, what is your strategy for improving homology-directed repair in stem cells? Do you have any methods that you recommend using?
Right. So to improve homology-directed repair, there's actually been quite a bit of literature on this because it is an area of active investigation. For what I have tested and find to be the most useful is in design of the donor DNA template. And so, as I mentioned, we incorporated some synonymous mutations into that donor DNA template, which do not change the way that the protein is going to be translated, but do alter the underlying DNA sequence. And this helps to prevent Cas9 and the guide RNA from reannealing, cutting, and generating an indel at that location. But I know that there are many groups looking at alternative DNA donor templates that might act with higher efficacy, such as adeno-associated viral DNA templates, which seem to really increase homology-directed repair efficiency. Alternatively, some people have used small molecules to shift the repair pathway away from using non-homologous end joining to using homology-directed repair. But again, similar to p53 inhibitors, global inhibition of non-homologous end joining or even promotion of homology-directed repair, we're not sure of the consequences at additional sites other than at the site you're editing. It doesn't seem to improve HDR all that much. And so, yeah, that area of the field is still actively investigated. And so hopefully something will be discovered soon.
I really want to thank everyone for attending, and I'd especially like to thank you, Ashley, for your time and your excellent presentation. And thanks again for everyone for attending.